Welcome to the next part of the lecture. This lecture has to do with the inadequacy of lexicon in clinical practice. So, for example, some bacterial commensals of the body, which shall be explained after I just repeat the first few lines which I just now mentioned at the previous lecture. Microbes capable of causing disease are routinely cultured from patients, and the decision to administer microbial therapy often depends on whether the microbe is judged to be a pathogen or colonizer. The medical literature contains different conclusions regarding the implications of recovering certain microorganisms from patients. For example, some bacteria commensals of the vagina have been associated with neonatal pneumonia, demonstrating, just a second, right? For example, some bacterial commensals of the vagina have been associated with neonatal pneumonia, demonstrating that the same organism can be a commensal in one host and a pathogen in another. The recovery of C. candida albicans from multiple sites presents the vexing question of whether it's a reflection of colonization or infection. Similarly, it is often very difficult to distinguish between colonization and infection with gram-negative microbes such as Pseudomonas spp are recovered from individuals on ventilator support and intensive care units. In patients with chronic obstructive lung disease, a variety of well-recognized bacterial pathogens can be continuously isolated from the lower airways even between disease exacerbations. Concerned that colonization can lead to higher rates of clinical disease and transmission to others underlies the practice of administering antimicrobial prophylaxis to contacts of individuals with N. meningitis and Aspergillus infection. On clinical medicine, the presence of an infection generally constitutes a requirement for therapy. In the absence of objective data indicative of infection, treatment of colonization is usually avoided due to its cost and risk of adverse reactions and of induction of microbial resistance. The standard definitions of infection and colonization are not helpful clinically in determining the significance of certain microbes from wounds and body sites that are normally sterile. At present, efforts to establish guidelines for the treatment of states of microbial colonization in the hospital setting are limited by an inadequate understanding of the pathogenesis of colonization and the aspects of host-microbe relationship that influence the development of infection after colonization. This has probably fostered empiricism in the management of infectious disease that may have significant deleterious consequences for patient care and increase emergence of resistant strains. The use of damage framework to clarify lexicon. Changes in the epidemiology of infectious disease, including an increased prevalence of emerging and nosocomial infections, increased number of hosts with immune impairment, and new scientific information about microbial pathogenesis have rendered parts of the lexicon inadequate and in need of revision. I repeat, and in need of a revision. So the absence of a unified framework to serve as a theoretical foundation for the lexicon has resulted in the persistent use of terms that emphasize differences between microbes and specific microbial attributes at the expense of common themes. A major consequence has been fragmented in the field such that the disciplines of bacteriology, mycology, parasitology, and virology are increasingly insular despite the fact that all study similar questions. In our view, this problem may be ameliorated by an integrated theory of microbial pathogenesis that considers the contributions of both hosts and pathogens in this process. Our first approach to this conundrum was to introduce the concept that host damage is the relevant outcome in host microbe interactions and to propose a pathogen classification scheme based on the ability of a microbe to cause damage as a function of the host's immune response. That framework grouped microbes based on the ability to inflict damage as a function of the host response, irrespective of their phylogenetic derivation, by focusing on damage instead of pathogen or host. Common themes in microbial pathogenesis became more apparent. The same framework can be used to clarify the lexicon of microbial pathogenesis. Integrated lexicon to describe host-pathogen interaction. right on the topic of integrated lexicon to describe host-pathogen interaction. Excuse me. We propose that the outcome of host-pathogenic interaction is determined by the nature of the damage that results from the host-microbe relationship. This concept is based upon three assumptions. First, the acquisition of a microbe by a host can result in some form of damage to host tissues that elicit such a microbe-specific immune response. Second, except for congenitally acquired microbes, mammalian hosts are born without a significant microbial burden. And the initial acquisition of microbial flora therefore results from infection. Third, the amount and type of damage that occurs during the initial and/or ongoing host-microbe relationship determines the outcome of the host-microbe relationship. This view is consistent with the danger hypothesis proposed by Metzinger and others that immune responses arise from the detection of danger signals produced by microbes or infected tissues. Thus, we propose that host damage is often a requirement for the induction of a pathogen-specific immune response, and that constancy, type, and magnitude of damage that ensues should form the basis of the lexicon of microbial pathogenesis. So this, what you see here, is the flowchart. Host microbe. Okay. We have a little bit of a continuation to be done from the inadequacy of the lexicon in clinical practice. So in clinical medicine, the presence of an infection generally constitutes a requirement of therapy, right? So the absence of objective data indicative of infection, treatment of colonization is usually avoided due to its cost and risk of adverse reaction and of induction of microbial resistance. The standard definitions of infection and colonization are not helpful clinically in determining the significance of the isolation of certain microbes from wounds and body sites that are normally sterile. At present, efforts to establish guidelines for the treatment of states of microbial colonization in the hospital setting are limited by inadequate understanding of the pathogenesis of colonization and the aspects of the host-microbe relationship that influence the development of infection after colonization. This has probably fostered empiricism in the management of infectious disease that may have significant deleterious consequences for patient care and increase the emergence of resistant strains. Okay, the use of damage framework to clarify lexicon is the next portion. Changes in the epidemiology of infectious diseases, including an increased prevalence of emerging and nosocomial infections, increased number of hosts with immune impairment, and new basic scientific information about microbial pathogenesis have rendered parts of the lexicon inadequate and in need of revision. The absence of a unified framework to serve as a theoretical foundation for the lexicon has resulted in the persistent use of terms that emphasize differences between microbes and specific microbial attributes at the expense of common themes. A major consequence has been fragmentation in the field such that the disciplines of bacteriology, mycology, parasitology, and virology are increasingly insular, despite the fact that all study similar questions. In our view, this problem may be ameliorated by an integrated theory of microbial pathogenesis that considers the contributions of both host and pathogen in this process. The first approach to this conundrum was to introduce the concept that host damage is the relevant outcome in host-microbe interactions, and to propose a pathogen classification based on the ability of a microbe to cause damage as a function of the host immune response. So basically it should be, that's right, a definition based on the ability of a microbe to cause damage as a function of the host's immune response is the standard classification, I'd say. And aspects related on that. And that framework grouped microbes based on the ability to inflict damage as a function of the host response, irrespective of the phylogenetic derivation, by focusing on damage instead of pathogen or host. Common themes in microbial pathogenesis became more apparent. The same framework can be used to clarify the lexicon of microbial pathogenesis. The next is integrated lexicon to describe host-pathogen interaction. Now, It's proposed that the outcome of a host-pathogen interaction is determined by the nature of the damage that results from the host-microbe relationship. This concept is based upon three assumptions. First, the acquisition of a microbe by a host can result in some form of damage to the host tissues that elicits a microbe-specific immune response. Second would be, except for congenitally acquired microbes, mammalian hosts are born without a significant microbial burden, and the initial acquisition of microbial flora therefore results from infection. Third would be the amount and type of damage that occurs during the initial and/or ongoing host-microbe relationship determines the outcome of the host-microbe relationship. Right? The amount or type of damage that occurs during the initial and/or ongoing host-microbe relationship determines the outcome of the host-microbe relationship. This view is consistent with the danger hypothesis proposed by Matzinger and others, that immune response arose from the detection of danger signals produced by microbes in infected tissues. So, we propose that host damage is often a requirement for the induction of a pathogenic specific immune response, and that the constancy, type, and magnitude of damage that ensues should form the basis of the lexicon of microbial pathogenesis. We stopped last year. We last stopped at this flowchart, and the host microbe contact, infection, commensalism, and symbiosis, death, elimination, colonization, etc. All shown here. Please take a screenshot of this and make your references. Outcome of the host microbe interaction in the context of damage framework proposed in reference number 7. The various states shown in this figure defined in table 2 double-headed arrows indicate conditions where there may be variable amounts of damage. Single-headed arrows indicate the following. Acquisition of a microbe can be followed by elimination through physical defenses or immune mechanism. Acquisition of certain microbes results in damage and disease in certain hosts. Certain commensal microbes can cause disease if the state of commensalism is disturbed by immune impairment or alterations of the host. Right? And the state of colonization may be determined by immune response, for example, transient nasopharyngeal carriage of N. meningitis, S. pneumonia. The trigger for the immune response is not well understood but may occur after the damage threshold is reached. The state of colonization may lead to disease if sufficient damage ensues from the interaction. The damage may be host-mediated, pathogen-mediated, or both. The state of colonization may lead to a state of persistence, chronicity, and latency, whereby the immune response is unable to eradicate the infection despite continued damage. Latent M. tuberculosis or histoplasma capsulatum infection, tissue granuloma. G. An immune response or therapy may eradicate the infection, but this does not always terminate disease, because the damage may be reversible, example poliomyelitis, or continue through immunological mechanisms, example reactive arthropathies. It is if sufficient damage is incurred as a result of the host microbe interaction, death ensues, and persistent infection may reactivate and cause overt disease, example reactivation tuberculosis. Now, infection. The word infection is from the Latin word infiere, right, which means to die, stain, corrupt, or spoil, and has been associated with a disease since antiquity. There's a lot of references to 17, number 40, these are lines. Ancient usage of this term includes the idea that disease was caused by invisible agents that entered the body. However, whereas some authors consider an infection to be the outcome of a tissue invasion, others have used the word to include initial contact between parasite and host, and others have defined the term to include disease causation. Now, to define infection as the acquisition of microbe by host, eradication of the microbe by the host can occur at first contact, thus bypassing infection, by non-immune mechanisms and subsequently by immunologic mechanisms. Successful immune responses and antimicrobial therapy reduces the amount of continuing damage caused by the microbe to a level that is insignificant. Notably, eradication of a microbe may not eliminate clinical diseases since immunological damage to the host may persist following a successful antipathogen response, example rheumatic heart disease following streptococcal pharyngitis, and reactive arthritis following gastrointestinal infection with certain bacteria. The definition of infection proposed here avoids the confusion surrounding the synonymous use of infection and disease and is consistent with several of the definitions. Furthermore, the concept that clinical disease is associated with damage and the induction of an immune response is consistent with the historical concept of immunologic proof of causation and applies to different types of microbes, and to the currently accepted paradigm that immune cells must interact directly with microbes or the antigens to produce an antigen-specific response. Table 2. Proposed revisions of terminology of microbial pathogenesis in the context of the damage framework. Term is carrier state, revised definition synonymous with colonization. Chronicity synonymous with persistence. Colonization, a state of infection that results in a continuum of damage from none to great, with the latter leading to the induction of host responses that could eliminate or retain the microbe or progress to chronicity or disease. For organisms that induce no damage during infection, this state is indistinguishable from commensalism. And commensal, a microbe that induces either no damage or clinically inapparent damage after primary infection, a state that is thought to be established early in life. Commensalism, a state of infection that results in either no damage or clinically inapparent damage to the host, though it can elicit an immune response. Damage, the interruption of a normal tissue structure and or function of the host that applies at the cellular tissue and organ levels. And necrosis, apoptosis, mutation, synaptic blockage, and malignant transformation are examples of damage at the cellular level. Granulomatous inflammation, fibrosis, tumor are examples of damage at the tissue level. Ductal obstruction is an example of damage at the organ level. Presence of a microbe-specific immune response may be indicative of a heretofore unrecognized manifestation of damage. Elimination. Removal of the microbe from the boundaries of the host by either physical factors, interference by host flora and an immune response, or therapy. Infection, acquisition of a microbe by host. Most infections are followed by multiplication of the microbe in the host, but this is not universal because some helminth infections can involve a single organism that does not replicate in the host. Infectious disease, the clinical manifestation of damage that results from a host-microbe interaction. Latency, synonymous with persistence. This term is often used to describe infections that are asymptomatic over long periods of time but can evolve into overt disease. Persistence, a state of infection in which the host response does not eliminate the microbe, resulting in continued damage over time. Persistence may evolve into overt disease depending on the balance of the host-microbe interaction. Pathogen, a microbe capable of causing host damage as defined in reference. Symbiosis and mutualism, a state of infection whereby both the host and the microbe benefit as a consequence of infection. Okay, now going to the word commensalism. The word commensalism, commensal, from the Latin roots com, meaning with mensa, meaning table, so, with table, and ao, meaning pertaining to u, it can be translated eating at the same table. Our suggestions suggest revision to the definition of commensal is in the spirit of the original meaning of the word. Commensalism is defined as a host-microbial interaction that does not result in perceptible ongoing or persistent damage. However, it is notable that this may not be the initial acquisition of commensal organisms may elicit damage For example, Escherichia coli is acquired shortly after birth, and this counter now the suggested revision to the definition of commensal is in the spirit of the original meaning of the word commensalism. So commensalism is defined as a host microbial interaction that does not result in a perceptible, ongoing, and or persistent host damage. However, it is notable that this may not be absolute, since the initial acquisition of commensal organisms may elicit damage in some hosts. For example, Escherichia coli is acquired shortly after birth, and this encounter places some infants at risk for E. coli meningitis during the first month of life. Similarly, symptomatic infections with Candida albicans can occur in the first year of life. Microbes that establish themselves early in life encounter an immature immune system that may be unable to mount effective responses. The latter raises the possibility that immunologic immaturity may facilitate the establishment of a commensal state, but more work is needed to examine the validity of this concept. Nevertheless, the microbes that comprise the normal microflora of the oral, respiratory, and gastrointestinal tracts provide important stimuli for the development of immunity. Commensal microbes are antigenic and can elicit antibody responses. It is not known whether these immune responses reflect the occurrence of an unidentified mode or form of damage to the host. Commensals also synthesize metabolites that are essential nutrients for the host and protect the host by physically and metabolically preventing the acquisition and establishment of more pathogenic microorganisms. In view of the fact that the endogenous microbial flora plays a protective role against more pathogenic microbes, conditions that compromise the viability of this flora may damage the host, as evidenced by the observation that disruption of the endogenous microflora by antimicrobials predisposes to certain infectious disease. Hence, the endogenous microflora, collectively called commensal, is acquired by infection, stimulates the immune system upon acquisition, and plays a beneficial role throughout life. Interestingly, it may be difficult to distinguish host and microbe, as illustrated by the fact that the DNA of endogenous retroviruses comprises 1% of the human genome, human body, 1% of the human genome. In fact, upon noting that there are 10 raised to 13 cells in the human body and 10 raised to 14 to 10 raised to 15 individual microbes, Heisenberg profoundly asked the question, who parasitizes whom? Right? There's 10 raised to 13 cells of the human body and 10 raised to 14 to 15 individual microbes. Who parasitizes whom? It was asked by Heisenberg. Colonization, persistence, and disease. Right, so the word disease derives from Old French and originally implied a departure from normal or easy living. Here we define disease as a clinical manifestation of damage that results from host-microbe interaction. Colonization is defined as a state in which the microbe may be present in the host for a variable duration of time. In a setting in which the level of damage is insignificant, there may be no distinction between commensalism and colonization. However, in keeping with the historical definitions, the establishment of the commensal state generally occurs early in life, when the damage associated with the prolonged states of colonization induces a new state in the host, example, a granuloma following infection with Mycobacterium and tuberculosis. The outcome is persistence. Thus, commensalism, colonization, and persistence are separate outcomes of infection, but they are potentially linked and continuously based on the induction of damage. Therefore, the damage experienced by the host during colonization is part of a continuum which spans from none as with that induced by a commensal to as significant as with that induced by a pathogen. Colonization is a state characterized by a microbial replication that may induce host response and trigger a microbe-specific immune response, which in turn could eradicate or contain the microbe. If the host immune response, antimicrobial therapy, and a vaccination succeeds in eradicating the microbe, the state of colonization is eliminated. If the microbe is not eliminated, a state of persistence may ensue. Progressive damage that results from this state may lead to disease and death. Thus, colonization and its modified versions, namely persistence and disease, represent the continued presence of microbes in the host with a variable degree of but continued host damage. Synthesis of revised lexicon. We propose that the outcomes in infection represent a continuum and that the occurrence of one versus another is a result of an interplay between host and microbial factors for a particular microbe and a particular host. This interplay permits some microbes to become ensouled in some hosts but to cause disease in others. Therefore, as discussed in our previous review, the attribute of microbial virulence and the distinction between pathogens and non-pathogens are critically dependent on host factors. Here, we reason that the outcome for an infection with an organism is also a function of host-microbe interaction. For example, C. albicans is a commensal in normal individuals with intact endogenous flora, but a pathogen in some immunosuppressed patients. Example, those with an immune disturbance, those receiving antibiotics. Example, those with a microflora disturbance, and neonates who have an immature gastrointestinal tracts and have not yet established the endogenous microbial flora. For example, immature hosts. Consistent with this view, it has been proposed that immunological function determines whether contact with C. albicans results in clearance, colonization, or candidiasis. Notably, the proposed definitions in Table 2 are consistent with most, if not all, of that many definitions already found in the literature. And the definition here of commensal is similar to that of White and Timbray, and the word damage is already part of many definitions. So this review represents an effort to integrate these terms into a unified framework based on the damage response hypothesis as put forth previously, to produce a simplified lexicon centered upon the outcome of host-microbial interaction rather than on distinctions between pathogens and non-pathogens. This approach avoids the current problem of definitions that are dependent upon specific microbial or host characteristics and often require qualifications because of the inherent variability among the participants in the host-microbe interactions. Thus, an advantage of the proposed lexicon may be reduced ambiguity. This and the previous articles arose directly from various experiences of teaching microbial pathogenesis. This is as said by scientists to graduate and medical students. So, it is hoped that the proposed modifications to lexicon will stimulate discussion, experimental support, or refute existing concepts. Current measures of damage rely on mortality, tissue destructions, clinical disease, or may be too sensitive to characterize host pathogen interactions that lead to colonization and chronicity. Right. We're moving on to the subject of cell biology of infection, right? So, we've just seen that the pathogens constitute a diverse set of agents. There are corresponding diverse ranges of mechanisms by which pathogens cause diseases, but the survival and success of all pathogens require that they colonize the host, reach an appropriate niche, avoid host defenses, replicate and exit the infected host to spread to an uninfected one. In this section, it is inevitable to examine the common strategies used by many pathogens to accomplish these tasks. So the first topic would be pathogens cross protective barriers to colonize the host. The first step in infection is for the pathogen to colonize the host. Most parts of the human body are well protected from the environment by a thick and fairly tough covering of skin. The protective boundaries in some other human tissues, eyes, nasal passage and respiratory tract, mouth and digestive tract, urinary tract, and female genital tract are less robust. For example, in the lungs and small intestines, where oxygen and nutrients respectively are absorbed from the environment, the barrier is just a single monolayer of epithelial cells. Skin and many other barrier epithelial surfaces are usually densely populated by normal flora. Some bacterial and fungal pathogens also colonize these surfaces and attempt to outcompete the normal flora, but most of them, as well as all viruses, avoid such competition by crossing these barriers to gain access to unoccupied niches within the host. Wounds and barrier epithelia, including the skin, allow pathogens direct access to the interior of the host. This avenue of entry requires little in the way of specialization on the part of the pathogen. Indeed, many members of the normal flora can cause serious illness if they enter through such wounds. Anaerobic bacteria of genus Bacteroides, for example, are carried as harmless flora or at very high density in the large intestine, but they can cause life-threatening peritonitis if they enter the peritoneal cavity through a perforation in the intestine caused by trauma, surgery, or infection in the intestinal wall. Staphylococcus from the skin or nose, or Streptococcus from the throat and mouth are all responsible for many serious infections resulting from bacteria, from breaches in epithelial barriers. Dedicated pathogens, however, need not wait for a well-timed wound to allow them access to their host. A particularly efficient way for a pathogen to cross the back skin is to catch a ride in the saliva of a biting insect. Many arthropods nourish themselves by sucking blood, and a diverse group of bacteria, viruses, and protozoa have developed the ability to survive in the arthropod so that they can use these biting animals as vectors to spread from one mammalian host to another. Interestingly written as to catch a ride in the saliva of a biting insect. Yeah, to catch a ride in the narrative, or whether it's physical or literary, to get into the next phase or media and get a continuity. Right, okay. So, and as discussed earlier, the plasmodium protozoan that causes malaria develops through several forms in its life cycle, including some that are specialized for survival in a human and some that are specialized for survival in a mosquito. So let's have a look at 2510. So that shows please take a screenshot as you see it, right? And there's the details on this. Okay, that's it about that. Excuse me. Viruses that are spread by insect bites include the causative agents for several types of hemorrhagic fever, including yellow fever and dengue fever, as well as the causative agents for many kinds of viral encephalitis, inflammation of the brain. All these viruses have acquired the ability to replicate in both insect cells and mammalian cells, as is required for a virus to be transmitted by an insect vector. Blood-borne viruses such as the HIV that are not capable of replicating in insect cells are rarely, if ever, spread from insect to human. The efficient spread of a pathogen via an insect vector requires the individual insects. Insects consume blood meals from numerous mammalian hosts. In a few striking cases, the pathogen appears to alter the behavior of the insect so that its transmission is more likely. Like most animals, the tsetse fly, whose bite spreads the protozoan parasite Trypanosoma brucei, which causes sleeping sickness in Africa, stops eating when it is satiated, but tsetse flies carry trypanosomes bite much more frequently and ingest more blood than do uninfected flies. The presence of trypanosomes impairs the function of the insect mechanoreceptors that measures blood flow through the gullet to assess the fullness of the stomach, effectively fooling the tsetse fly into thinking that it is still hungry. The bacterium Yersinia pestis, which causes bubonic plague, uses a different mechanism to ensure that a flea carrying it bites repeatedly. It multiplies in the flea's foregut to form aggregated masses that eventually enlarge and physically block the digestive tract. The insect is then unable to feed normally and begins to starve. During repeated attempts to satisfy its appetite, some of the bacteria in the foregut are flushed into the bite site, thus transmitting plague to a new host. That's interesting there, right? Figure 2518. The spread of plague. This micrograph shows the digestive tract dissected from a flea that had dined about 2 weeks previously on the blood of an animal infected with plague bacterium, Yersinia pestis. Have a look at this. Okay. The bacterium multiplied in the flea gut to produce large adhesive aggregates, cohesive aggregates, indicated by the red arrows. Right, Yersinia. The bacterial mass on the left is occluding the passage between the esophagus and the midgut. This type of blockage prevents a flea from digesting its blood meals, thereby causing it to bite repeatedly, disseminating the infection. Right? Let's repeat that part. The bacteria multiply in the flea gut to produce large cohesive aggregates, indicated by the red arrows. That you just saw it. The bacterial mass on the left, have a look, on the left, is occluding the passage between the esophagus and the midgut. This type of blockage prevents a flea from digesting its blood meals, right, thereby causing it to bite repeatedly, disseminating the infection. Yeah. Going back to subject matter proper, that was it about that figure. And then about pathogens that colonize epithelium must avoid clearance by the host. Hitching a ride through the skin on an insect proboscis is just one strategy that pathogens use to pass through the initial barriers of host defense. Whereas many barrier zones such as the skin, mouth, and large intestine are densely populated by normal flora, others including the lower lung, the small intestine, and the bladder are normally kept nearly sterile. Despite relatively direct access to the environment, the epithelium in these zones actively resists bacterial colonization. The respiratory epithelium is covered with a layer of protective mucus and the coordinated beating of cilia. Sweeps the mucus and trapped bacteria and debris up and out of the lung. The epithelial lining the bladder and the upper gastrointestinal tract also have a thick layer of mucus, and these organs are periodically flushed by micturition and peristalsis, respectively, to wash away undesirable microbes, the pathogenic bacteria, excuse me, and parasites that infect these epithelial surfaces are specific mechanisms for overcoming these host cleaning mechanisms. Those that infect the urinary tract, for example, resist the washing action of urine by adhering tightly to the bladder epithelium via specific adhesions. Proteins or protein complexes that recognize and bind to host cell surface molecules. An important group of adhesions in uropathogenic E. coli strains are components of the P. pili that help the bacteria infect the kidney. These surfaces projections can be several micrometers long and are thus able to span the thickness of the protective mucus layer. At the tip of each pilus is a protein that binds tightly to a particular disaccharide linked to a glycolipid that is found on the surface of cells in the bladder and kidney. And figure 2519, an uropathogenic E. coli and P. pili scanning electron micrograph of uropathogenic E. coli, a common cause of bladder and kidney infections, and attached to the surface of epithelial cells in the bladder of an infected mouse. Have a look at the picture. This is one, the bacteria there, E. coli and P. pili. Right, a close-up view of the bacteria shows the pili on the surface. Right. And one of the hardest organs for a microbe to colonize is the stomach. Besides peristaltic washing and protection by a thicker layer of mucus, the stomach is filled with acid. Average pH is 2. So this extreme environment is lethal to almost all bacteria ingested in food. Nonetheless, it is colonized by the hardy and enterprising bacterium, Helicobacter pylori. which was recognized only recently as a major causative agent in stomach ulcers and possibly stomach cancer. Although the older treatments for ulcers, acid-reducing drugs and bland diets, still used to reduce inflammation, a short and relatively cheap course of antibiotics can now effectively cure a patient of recurrent stomach ulcers. The hypothesis that stomach ulcers could be caused by a persistent bacterial infection of the stomach lining initially met with considerable skepticism. The point was finally proved by the young Australian doctor who made the initial discovery. He drank a flask of pure culture of H. pylori and developed a typical ulcer. One way that H. pylori survives in the stomach is by producing the enzyme urease, which converts urea to ammonia and carbon dioxide. In this way, the bacterium surrounds itself with a layer of ammonia, which neutralizes stomach acid in its immediate vicinity. The bacteria also express at least 5 types of adhesions, which enable them to adhere to the stomach epithelium, and they produce several cytotoxins that destroy the stomach epithelial cells, creating painful ulcers. The disease resulting chronic inflammation promotes cell proliferation and thus predisposes the infected individual to stomach cancer. Excuse me. A more extreme example of active colonization is provided by Bordetella pertussis, the bacterium that causes whooping cough. The first step in a B. pertussis infection is colonization of the respiratory epithelium. The bacteria circumvent the normal clearance mechanism, the mucociliary escalator described in another chapter, by binding tightly to the surface of ciliated cells and multiplying on them. B. pertussis expresses at least 4 types of adhesions that bind tightly to particular glycolipids on the ciliated cells. The adherent bacteria produce various toxins which eventually kill the ciliated cells, compromising the host's ability to clear the infection. The most familiar of these is pertussis toxin, which, like cholera toxin, is an ADP ribosylating enzyme. It ADP ribosylates the subunit of the G protein G1, causing deregulation of the host cell adenylyl cyclase and overproduction of cyclic AMP. Not content with this, B. pertussis also produces an adenylyl cyclase of its own, which is inactive unless bound to the eukaryotic calcium-binding protein calmodulin. The bacterially produced enzyme is therefore active only in the cytoplasm of a eukaryotic cell. Although both B. pertussis and V. cholera have this similar effect of drastically raising CAMP levels in the host cells to which they adhere, the symptoms of the disease are very different because the two bacteria colonize different sites in the host. B. pertussis colonizes the respiratory tract and causes paroxysmal coughing, whereas V. cholerae colonizes the gut and causes watery diarrhea. Not all examples of specific colonization require that the bacterium express adhesions that bind to host cell glycolipids or proteins. Enteropathogenic E. coli, which causes diarrhea in young children, instead uses a type 3 secretion system to deliver its own bacterium-expressed receptor protein called TIR into its host cell. After TIR is inserted into the host cell membrane, a bacteria surface protein binds to the extracellular domain of TIR, triggering a remarkable series of events inside the host cell. First, the TIR receptor protein is phosphorylated on tyrosine residues by a host protein, tyrosine kinase. Unlike eukaryotic cells, bacteria generally do not phosphorylate tyrosine residues, yet TIR contains a peptide domain that is a specific recognition motif for a eukaryotic tyrosine kinase. The phosphorylated TIR then is thought to recruit a member of the RHO family of small GTPases, which promotes actin polymerization through a series of intermediate steps, which were already discussed in one of the chapters here. The polymerized actin forms a unique cell surface protrusion called a pedesto that pushes the tightly adherent bacteria up about 10 micrometers from the host cell surface. Figures 2520b. Let's see that. take a screenshot. I think it should explain things better to you. Yeah. So interaction of enteropathogenic E. coli, EPEC, with host cells. When EPEC contacts an epithelial cell in the lining of the human gut, it delivers a bacterial protein, TIR, into the host cell through a type 3 secretion system. TIR then inserts into into the plasma membrane of the host cell, where it functions as a receptor for bacterial adhesion in timin. The intracellular domain of TIR is phosphorylated on a tyrosine residue by a host cell protein tyrosine kinase. Phosphorylated tyrosine then TIR and probably recruits a Rho family GDPase, which triggers actin polymerization. It also recruits other host cell cytoskeletal factors that interact with the actin. Consequently, a bundle of actin elements is assembled under the filaments is assembled underneath the bacterium to form an actin pedestal, EPEC on a pedestal. In this fluorescence micrograph, the DNA of the epoch and host cell are labeled in blue, TIR protein is labeled in green, and host cell actin filaments are labeled in red. So the insert shows a close-up view of two of the bacteria on pedestals. Right, that's about the diagram there. Going on to the subject matter proper. These examples of the host colonization illustrate the importance of host pathogen communication in the infection process. Pathogenic organisms have acquired genes that encode proteins that interact specifically with particular molecules of the host cells. In some cases, such as the B-pertussis adenylyl cyclase, an ancestor of the pathogen may have acquired the gene from its host, whereas in others such as TIR, random mutation may have given rise to protein motifs that are recognized by a eukaryotic protein partner. Intracellular pathogens have mechanisms for both entering and leaving host cells. Many pathogens, including V. cholerae and B. pertussis, infect their host without entering host cells. Others, however, including all viruses and many bacteria and protozoa, are intracellular pathogens. Their preferred niche for replication and survival is within the cytoplasm or intracellular compartment of particular host cells. This strategy has several advantages. The pathogens are not class accessible to antibodies, and they are not easy targets for phagocytic cells. This lifestyle, however, does require that the pathogen develop mechanisms for entering host cells, for finding a suitable subcellular niche where it can replicate, and for exiting the infected cell to spread the infection. In the remainder of this section, we consider some of the myriad ways that individual intracellular pathogens exploit and modify host cell biology to satisfy these requirements. Viruses bind to molecules displayed on the host cell surface is the next topic. The first step for any intracellular pathogen is to bind to the surface of the host target cell. For viruses, binding is accomplished through the association of a viral surface protein with a specific receptor on the host cell surface. Of course, no cell host cell receptor evolved for the sole purpose of allowing a pathogen to bind to these receptors. All have other functions. First, such virus receptor identified was the E. coli surface protein that allows the bacteriophage lambda to bind to the bacterium. Its normal function is a transport protein responsible for the uptake of maltose. Viruses that infect animal cells generally use cell surface receptor molecules that are either very abundant, such as sialic acid containing oligosaccharides, which are used by the influenza virus, or uniquely found on those cell types in which the virus can replicate, such as the nerve growth factor receptor, the nicotinic acetylcholine receptor, or the cell adhesion protein NCAM, all of which are used by the rabies virus to specifically infect neurons. Often a single type of receptor is used by many types of virus, and some viruses can use several different receptors. Moreover, different viruses that infect the same cell type may each use a different receptor. Hepatitis, for example, is caused by at least 6 viruses, all of which preferentially replicate in liver cells. Receptors for 4 of the hepatitis virus have been identified. They are all different. Receptors need not be proteins. Herpes simplex virus, for example, binds to heparin sulfate proteoglycans through specific viral membrane proteins. Frequently, viruses require both a primary receptor and a secondary co-receptor for efficient attachment and entry into host cells. An important example is HIV. Its primary receptor, CD4, a protein involved in immune recognition, which is found at the surface of many T cells and macrophages. Viral entry also requires the presence for an alpha chemokines, depending on the beta chemokines, or a CXCR4 receptor for alpha chemokines, depending on the particular variant of the virus. Macrophages are susceptible only to HIV variants that use CCR5 for entry, whereas T cells are more efficiently infected by variants that use CXCR4. The viruses that are found within the first few months after HIV infection almost invariably require CCR5, which presumably explains why individuals who carry a defective CCR5 gene are not susceptible to HIV infection. When you read that, CCR5, which is defective, are not susceptible to HIV infection. Viruses may either switch to use the CXCR4 co-receptor or adapt to use both co-receptors. In this way, the virus can change the cell types it infects as the disease progresses. Pretty tricky virus. Never knew a virus could be that intelligent. Great. Receptor and co-receptors for HIV. All strains of HIV require CD4 as a primary receptor. Early in an infection, most of the viruses use CCR5 as a co-receptor, allowing them to infect macrophages and the precursors, monocytes, as the infection progresses. Viruses enter host cells by membrane fusion, pore formation, or membrane disruption. After recognition and attachment to the host cell surface, the virus must next enter the host cell and release its nucleic acid genome from its protective protein coat or lipid envelope. In most cases, the liberated nucleic acid remains complexed with some viral proteins. Enveloped viruses enter the host cell by fusing either with the plasma membrane or with the endosomal membrane following endocytosis. Fusion is thought to proceed via a mechanism similar to snare-mediated fusion of vesicles during normal intracellular vesicular traffic. For viruses uncoating strategies. Some enveloped viruses, such as HIV, fuse directly with the host cell plasma membrane to release their capsid into the cytosol. Other enveloped viruses, such as influenza virus, first bind to cell surface. Fusion is regulated both to ensure that virus particles fuse only with the appropriate host cell membrane and to prevent virus particles from fusing with one another. For viruses such as HIV that fuse at neutral pH at the plasma membrane, binding to receptors or co-receptors using triggers a conformational change in the viral envelope protein to expose a normal buried fusion peptide. This is in figure 13.16. Right. The other enveloped virus, such as influenza, postpone fusion until after endocytosis. In the case, it is frequently the acid environment in the early endosome that triggers the conformational change in a viral surface protein that exposes the fusion peptide. The H pumped into the early endosome enters the proton, enters the influenza particle through an ion channel and triggers the uncoating of the viral nucleic acid, which is directly released into the cytosol as the virus fuses with the endosomal membrane. For some viruses, uncoating occurs after release into the cytosol. In the case of semiliki-forest virus, for example, the binding of host ribosomes to the capsid causes the capsid proteins to separate from the viral genome. The entry strategy used by influenza virus, the globular heads of the viral hemagglutinin HA mediate binding of the virus to sialic acid containing cell surface receptors. The virus receptors complexes are endocytosed and in the acidic environment. It is more difficult to envision how non-enveloped viruses enter host cells, as it is not obvious how large assemblies of protein and nucleic acids can cross the plasma or endosomal membrane, where the entry mechanism is understood. Non-enveloped viruses generally either form a pore in the cell membrane to deliver the viral genome into the cytoplasm, or they disrupt the endosomal membrane after endocytosis. Now, polioviruses uses the first strategy. Binding a poliovirus to its receptor triggers both receptor-mediated endocytosis and a conformational change in the viral particle. The conformational change exposes a hydrophobic projection on one of the capsid proteins, which apparently inserts into the endosomal membrane to form a pore. The viral genome then enters the